Welcome to another Deep Dives. Today we're going to be talking about something you might not think about too much, but it's pretty important. Impurities in drugs.
Right.
And we're not just talking about like pharmaceuticals,
Mhm.
like, the stuff you get from the pharmacy with a prescription.
Yeah.
We're talking about, cannabis extracts, psilocybin extracts, even herbal supplements.
Yeah, all of that.
Yeah.
Anything that you're putting in your body, basically, that's going to
Okay.
have some sort of effect.
So why is this so important? Like, what's the big deal about a little bit of impurities?
Well, it's interesting because even tiny amounts of these impurities can have a really significant impact on how safe a drug is and how well it works.
Mm-hmm.
It's kind of like, if you have like a glass of water and you put a drop of poison in it,
Right.
it doesn't take much to make the whole thing dangerous.
Right, so a little bit goes a long way.
Exactly.
So we're going to be diving deep into how these kind of hidden, I'm going to call them gremlins. How do we identify them? How do we categorize them? And how do we control them to make sure that the medications we take and the substances we take are safe and effective?
Yeah, and, we'll be talking about this in the context of, pharmaceuticals, cannabis, psilocybin.
Right.
We'll use those as examples.
Yeah, to give people some real-world context.
Exactly.
Now, we're also going to be exploring some ICH guidelines.
Yeah.
I know those sound kind of boring, but it's sort of like a rule book for the pharmaceutical industry.
Yeah, it's like the gold standard. Like if you are producing these types of products,
Right.
you want to know these guidelines inside and out.
Exactly. So these guidelines are Q3A through Q3E.
Uh-huh.
And they cover a lot of stuff from how to identify impurities and what different kinds there are, to even like setting limits for impurities that maybe we don't know that much about.
Yeah, it's a really comprehensive framework.
So let's get down to the nitty-gritty. How do scientists even begin to find these impurities? It's not like you can just, look under a microscope and be like, oh, there's an impurity.
Right. Yeah, it's not that simple.
So what do they do?
Well, they have to rely on some pretty sophisticated analytical techniques. One of the most powerful tools that they use is called NMR spectroscopy.
Okay.
And this allows them to figure out the structure of even like really tiny amounts of these compounds that we don't want in there.
Wow. So they can look at it on like a molecular level.
Yeah, basically it's like taking a fingerprint of the substance and then looking for anything that looks out of place.
That's really cool.
Yeah.
So one thing we came across in our research is this concept of purge factors.
Uh-huh.
What are those and why do we care about them?
So purge factors are basically a way to measure how well a purification step gets rid of an impurity. So let's say, for example, you have a step in your process that removes 99% of a specific impurity.
Okay.
The purge factor for that step would be 100. So the higher the purge factor, the better that purification step is working.
So it's kind of like a measure of efficiency.
Exactly. And these calculations are really important because they let us know, is the step actually doing what it's supposed to be doing?
Right.
And ultimately, it all ties back to making sure that the final product is safe for patients.
So it's kind of like if you're filtering your water at home.
Yeah.
You want to make sure that filter is actually getting rid of the stuff you don't want in there.
Exactly. You don't want to be drinking contaminated water.
Yeah.
And you don't want to be taking medication that has something in it that could hurt you.
Right. So it's like purification, but on a much more, microscopic level.
Exactly.
Okay. So let's say you've identified an impurity. What happens next?
Well, the next step is to classify it.
Okay. How does that work?
So the ICH has this system for categorizing impurities.
Uh-huh.
And it's based on where they come from and how likely they are to cause harm. So you've got organic impurities, which are basically derived from living things, inorganic impurities, which come from non-living sources. And then you have residual solvents, which are left over from the manufacturing process and so on.
So it's like sorting your laundry. You get your whites, your darks, your delicates.
Yeah, you're grouping them based on their properties and their potential impact.
That's a good analogy.
Yeah, because just like you wouldn't want to wash your delicates with your jeans, you wouldn't want to treat all impurities the same way.
Right, they need special treatment.
Exactly.
Now, our sources actually specifically mentioned nitrosamine impurities as being particularly problematic.
Yeah.
What makes those so bad?
Well, nitrosamines like NDMA and NDEA are a class of compounds that can actually form unexpectedly in certain drug products.
Oh, wow. So even if everything seems fine initially, they can just pop up later.
Right. And it tends to happen during storage, which is what makes them so trippy. And the reason why they're so concerning is that they're known to cause cancer. Even in very small amounts, they can pose a real risk to human health.
So it's kind of like finding a rotten apple in a barrel that looked perfectly fine a few days ago.
Right. You got to stay vigilant.
Exactly. And those valsartan and ranitidine recalls a few years back, those were because of nitrosamines, right?
Yeah, they found NDMA in those products, which led to them being pulled from the market.
It's a good reminder that, you really can't be too careful when it comes to these things.
Exactly.
Now, speaking of things you don't want to find in your medicine, our sources highlighted some case studies involving unregulated cannabis and psilocybin products.
Yeah.
And these products were found to contain some pretty nasty contaminants.
Yeah, it's a real problem in unregulated markets.
What kind of things are we talking about here?
Well, it really runs the gamut, but you can find things like heavy metals, pesticides, microbial growth, and even leftover solvents from the extraction process.
So it's like you think you're getting this, natural product,
Yeah.
but it could be contaminated with all sorts of harmful stuff.
Exactly, and that can have serious consequences for your health, especially if you're already dealing with other medical conditions.
So it's buyer beware.
Absolutely. You really need to do your research and make sure you're getting these products from reputable sources.
Right, places that prioritize testing and quality control.
Exactly, because a cheap deal isn't worth risking your health.
That's a really good point.
And this actually brings us back to those ICH guidelines we talked about earlier.
Right.
They really are the foundation for ensuring that any substance we put in our bodies is safe and effective.
Okay, so let's break down these guidelines a little bit.
Right.
We've got Q3A through Q3E.
Mm-hmm.
Let's start with Q3A. What does that one cover?
So Q3A focuses specifically on impurities in new drug substances.
Okay.
So this is all about identifying, classifying, qualifying, and controlling those impurities from the very beginning of the drug development process.
So before it even gets to the point of being a finished product.
Exactly. It's like a roadmap for dealing with those unwanted compounds.
Yeah.
It covers everything from setting acceptable limits for those impurities to assessing the risk they might pose to patient safety.
It's like a comprehensive guide to making sure that those drug substances are as pure as possible.
Exactly.
Okay, so we've got the foundation laid with Q3A.
Mm-hmm.
What about Q3B? Where does that fit in?
Q3B zeroes in on residual solvents in those drug substances.
Residual solvent. So that's like the stuff that's left over from the manufacturing process.
Right. It's like when you're cleaning something and a little bit of the cleaning product stays behind.
Okay.
And the thing is, some of these solvents can be toxic.
Right, so we need to make sure we're not ingesting too much of them.
Exactly. That's why it's so important to keep their levels below those acceptable limits.
So Q3B is all about making sure that those leftover solvents don't end up posing a risk to patients.
Yeah, it's a crucial part of the safety puzzle.
Okay, what about Q3C? What does that one deal with?
So Q3C shifts the focus to impurities in the new drug product itself.
Okay, so that's like the final product that people actually end up taking.
Right, exactly. So now we're not just looking at the isolated drug substance, but the entire formulation, everything that goes into that final product.
Right, so it's a broader perspective.
It is, and it takes into account not only the impurities that might be present in the drug substance itself, but also anything that could be introduced during the process of making the final product, like during formulation or manufacturing.
So it's like a final quality check before that drug product hits the market.
That's a good way to think about it. And it also deals with things like the stability of the drug product and the potential for impurities to form over time.
Like those pesky nitrosamines we talked about.
Exactly.
Q3D, what's that all about?
Q3D is all about elemental impurities.
Elemental impurities, those sound a bit more serious.
Yeah, they can be. We're talking about things like heavy metals,
Huh.
which, as you know, can build up in the body and have really toxic effects, even in tiny amounts.
Right, so we really don't want those in our medications.
Yeah. Absolutely. So Q3D provides guidance on how to identify where those elemental impurities might be coming from, how to assess the risks they pose, and how to control their levels in those final drug products.
So it's like a safeguard against heavy metal contamination.
Exactly. It's all about minimizing those risks.
Okay, so we've covered Q3A, Q3B, Q3C, and Q3D. That leaves us with Q3E.
Right.
What does that final guideline focus on?
So Q3E is all about setting limits for impurities that haven't been qualified.
Unqualified impurities. You mean impurities that they don't know anything about?
Not necessarily. It refers to impurities that haven't been fully characterized or tested for safety.
Okay, so it's like, we know they're there, but we need more information.
Exactly. And so until we have that complete picture, Q3E helps to establish limits based on the best available knowledge at the time.
So it's a precautionary approach.
It is. It's all about erring on the side of caution and protecting public health.
I like that, erring on the side of caution.
It's always better to be safe than sorry.
So we've learned a lot about these ICH guidelines.
Yeah, it's a lot to take in.
It's really encouraging to know that there are these, pretty strict regulations in place
Uh-huh.
to make sure that the medications we take and the substances we use are safe and effective.
Right, and these guidelines aren't static either.
Oh, right.
They're constantly being reviewed and updated as new scientific knowledge emerges and new technologies become available.
So it's an ongoing process.
Exactly. It's a dynamic field that's always evolving to stay ahead of the curve and protect public health.
That makes me feel a lot better, knowing that someone's keeping an eye on all of this.
Absolutely. It's all about continuous improvement.
All right, so I think we've covered a lot of ground here in part 1 of our deep dive.
Yeah, we've laid a good foundation.
But before we move on to part 2,
Okay.
I want to ask you, dear listener, what has stood out to you so far?
Yeah, what's got you thinking?
What are you going to be paying more attention to now when it comes to the medications and substances you use?
We'll be back in a moment to delve deeper into this fascinating world of impurities and explore some of those cutting-edge technologies that are shaping the future of this field.
Stay tuned.
Welcome back to our deep dive on drug impurities.
I'm ready to dive back in.
Before we go any further, I just want to emphasize something we touched on earlier. Those ICH guidelines, Q3A through Q3E, they're not just, static documents sitting on a shelf somewhere.
Right.
They're constantly being reviewed and updated as science and technology advance.
So it's not a one-and-done situation.
Exactly. The goalposts are always moving as we learn more about impurities.
Makes sense.
And that brings us to some really exciting innovations that are happening in the field of impurity control. One area that's really transforming things is the development of what are called hyphenated techniques.
Hyphenated techniques. That sounds kind of intimidating.
Yeah, it's a mouthful, but it's actually a pretty simple concept.
Okay.
Basically, it involves combining 2 or more analytical methods to create a more powerful and precise tool.
So it's like getting a more complete picture.
Exactly. Imagine being able to examine a piece of evidence with a magnifying glass and a microscope at the same time.
You'd get a much more detailed view.
Right. You'd see things you wouldn't be able to see with just one or the other.
I'm starting to get it. So what are some examples of these hyphenated techniques?
Well, one common example is combining gas chromatography with mass spectrometry, or GCMS for short.
Okay.
Another one is liquid chromatography with mass spectrometry, or LCMS.
So you're basically pairing up different methods to get a more comprehensive analysis.
Exactly. And this allows scientists to not only identify impurities, but also measure how much of them is present with incredible accuracy.
So they can detect even the tiniest traces of those unwanted compounds.
Exactly. And they can also distinguish between compounds that are very similar in structure.
That sounds pretty crucial as we move towards more personalized medicine and more complex drug formulations.
Absolutely. As drugs become more sophisticated, so do the tools we need to analyze them.
Makes sense.
And speaking of sophisticated tools, artificial intelligence is starting to make its mark on the field of impurity control as well.
AI, like those self-driving cars everyone's talking about.
The very same AI and machine learning are being used to analyze vast amounts of data from those analytical instruments we talked about.
So it's like having a supercomputer helping to sift through all that information.
Exactly. And AI can help identify patterns and anomalies that might not be obvious to the human eye.
So it's like having a digital detective on the case.
That's a great way to put it. AI can potentially improve both the speed and accuracy of impurity detection.
That's pretty impressive.
And it can also be used to predict the likelihood of impurities forming during manufacturing.
So you can get ahead of the problem before it even happens.
Exactly. You can adjust the manufacturing process to minimize those risks.
That's some really powerful stuff.
It is. And as AI technology continues to evolve, we can expect even more groundbreaking applications in the field of impurity control.
So we've got all these incredible advancements in technology, but I imagine there are still some challenges when it comes to keeping those impurities in check.
Of course, there are always challenges, especially in a field that's constantly evolving. One of the biggest hurdles is keeping up with the ever-changing regulatory landscape.
Right, because those regulations are always trying to catch up to the latest scientific breakthroughs.
Exactly. And as new drugs and therapies emerge, new regulations and guidelines for impurity control need to follow suit.
It's like an ongoing race between science and regulation.
That's a good way to think about it. Another significant challenge is the increasing complexity of those drug formulations.
How so?
Well, as drugs become more targeted and personalized, the analytical methods used to ensure their safety and efficacy need to become even more sophisticated.
So it's like trying to solve a more intricate puzzle with the same set of tools.
Precisely. And developing those advanced methods can be both time-consuming and expensive.
I imagine it's a constant balancing act, pushing the boundaries of innovation while also making sure those new therapies are accessible and affordable.
It's a delicate balance for sure. And then, of course, there's the challenge of globalization.
How does that play into things?
Well, as drug manufacturing becomes more globalized, it becomes more difficult to ensure consistent quality control across different facilities and suppliers all over the world.
Right, so you've got different countries, different standards, different regulations.
Exactly, and that can make it tricky to guarantee that a drug product made in one country is of the same quality as one made in another country.
There's a real challenge to maintain those high standards on a global scale.
It requires a lot of communication, collaboration, and harmonization of standards across the board.
Makes sense. Okay, so we've got evolving regulations, increasingly complex drug formulations, and the added complexity of globalization. It sounds like a pretty tall order to stay on top of all that.
It's definitely a lot to juggle, but despite these challenges, the field of impurity control is constantly innovating and finding new ways to ensure the safety of the medications we rely on.
That's reassuring to hear. Now let's shift gears a bit and talk about how these concepts of impurity control apply to the real world.
We've mentioned cannabis and psilocybin a few times. Can you elaborate on the specific challenges of ensuring purity in those substances?
Certainly, cannabis, for instance, can be contaminated with a wide range of impurities depending on how it's grown and how those extracts are produced.
We've all heard those horror stories about contaminated cannabis products. It seems like a real minefield out there.
It can be, especially in unregulated markets without proper oversight. You can end up with cannabis products that contain heavy metals from the soil, pesticide residues, microbial growth, even residual solvents from the extraction process.
That's a pretty scary thought. You think you're getting this natural product, but you could be ingesting all sorts of harmful substances.
Exactly, and those contaminants can pose serious health risks, particularly for people with underlying medical conditions. So testing is absolutely essential, especially as cannabis becomes more widely used for medicinal purposes.
Absolutely. Reputable producers and dispensaries should have their products tested by independent third-party labs to make sure they meet those quality and safety standards.
So as consumers, we should be looking for those lab results to verify that what we're buying is actually clean.
Exactly. And the same goes for psilocybin.
What are some of the specific concerns with psilocybin?
Well, because psilocybin is often extracted from mushrooms that are grown in a variety of environments, contamination with heavy metals from the soil or pesticides used in cultivation can be a real concern.
So it's like those pesticides can travel all the way from the farm to the final product, even after all that processing.
It's a real possibility, which is why having those strict controls in place throughout the entire supply chain, from cultivation to extraction to purification, is absolutely critical. Every step needs to be carefully monitored and documented to ensure safety.
And are the testing methods for psilocybin impurities similar to the ones used for cannabis?
There's definitely some overlap, but there are also some unique challenges with psilocybin. For one thing, it's a much more potent substance than cannabis, so even smaller amounts of impurities can have a more significant impact.
So you need even more sensitive detection methods.
Exactly. You need to be able to find those impurities at extremely low levels. And because psilocybin research is still relatively new, there aren't as many standardized tests for impurities compared to more established pharmaceuticals.
That makes sense. As research expands, we can expect those testing standards to become more refined.
Absolutely. But for now, it's even more crucial to source your psilocybin from reputable suppliers who prioritize quality control and transparent testing.
So for listeners who might be considering using cannabis or psilocybin, what's the bottom line? What can they do to make sure they're getting safe and effective products?
The most important thing is to know your source. Always buy from reputable manufacturers and dispensaries who prioritize quality control and transparent testing. Look for products that have been tested by those independent third-party labs, and don't be afraid to ask for those lab results. It's your health we're talking about.
It's like doing your due diligence before making any purchase. You want to know where it came from, how it was made, and what's in it.
Exactly. And remember, if a deal seems too good to be true, it probably is. Be wary of products that make extravagant claims or are suspiciously cheap. Quality comes at a price. And when it comes to your health, that's a price worth paying.
Couldn't agree more. That's some really solid advice, and it highlights the importance of being an informed consumer, especially in these emerging markets now. Before we wrap things up with those cutting-edge technologies, I want to turn it back to our listeners.
Yeah, what's resonating with you so far? What's been the most surprising or intriguing aspect of this deep dive into the world of drug impurities? We'd love to hear your thoughts.
Welcome back to the final part of our deep dive into the world of drug impurities.
We've talked about identifying them, classifying them, and now it's time to explore the future of this field.
I'm excited to see what's on the horizon.
Well, one area where we're seeing some really groundbreaking advancements is in the development of analytical techniques that can detect impurities at even lower levels than ever before.
How low are we talking?
Think parts per billion or even parts per trillion.
Wow. That's like finding a single grain of sand on a beach.
Exactly.
Right.
It's an incredible level of sensitivity, and it's all thanks to some really innovative technologies.
Like what?
Well, one exciting development is the use of microfluidic devices.
Microfluidic devices. What are those?
They're basically like tiny labs on a chip.
Okay.
These devices allow scientists to do those complex chemical analyses we talked about, but on super tiny volumes of liquid.
So it's like shrinking down a whole laboratory.
Exactly, and it comes with a lot of advantages. For one, it reduces the amount of sample you need, which is great when you're dealing with rare or expensive substances.
That makes sense.
It also speeds up the whole analysis process, giving you faster results.
I bet that's crucial in situations where you need to identify a contamination issue quickly.
Absolutely. And because they're so small, microfluidic devices are portable too, you can take them out into the field for things like environmental testing.
It's like having a pocket-sized lab.
Exactly. Now, another technology that's showing a lot of promise is the use of biosensors.
Biosensors. What are those all about?
Well, a biosensor combines a biological component, like an enzyme or an antibody, with a transducer that converts a biological response into a measurable signal.
So it's like using nature's own detection system.
Exactly. Pretty ingenious, right?
It is. So what are the benefits of using biosensors?
Well, for one, they're often even more sensitive and selective than traditional methods, so they can detect even lower levels of impurities and differentiate between very similar compounds.
Wow. That sounds like a game changer for quality control and pharmaceutical manufacturing.
Absolutely. And because they're relatively inexpensive to produce, they have the potential to make high-quality testing more accessible, especially in resource-limited settings.
That's fantastic. So we've talked about these incredible advancements in detection technology, but what about preventing those impurities from forming in the first place?
That's a great question, and there are some exciting innovations happening on that front, too. One area that's generating a lot of buzz is the use of continuous manufacturing processes.
Continuous manufacturing. I've never heard of that. How is it different from traditional manufacturing?
Well, traditional pharmaceutical manufacturing is typically a batch process, meaning you produce a set amount, then you test it, and then you move on to the next batch.
Okay.
Continuous manufacturing, on the other hand, is more like a flow process.
Okay.
It's like having a conveyor belt that churns out a steady stream of product with constant monitoring and adjustment along the way.
So what are the benefits of this continuous approach?
Well, for one, it leads to much more consistent product quality because you're constantly monitoring and adjusting the process as you go. You can reduce the variability between batches, which in turn reduces the likelihood of those impurities forming.
So it's a more proactive approach to quality control.
Exactly, and it's also more efficient and generates less waste, making it a more sustainable approach overall.
Sounds like a win-win situation. Okay, so we've got all these amazing advances in both detection and prevention technologies, but I imagine there are still some challenges to overcome before we see the full impact of these innovations.
Of course, one of the biggest challenges is the need for regulatory frameworks that can keep up with the rapid pace of technological development.
It's that constant dance between science and regulation.
Exactly. Regulatory agencies need to have the resources and expertise to properly evaluate these new technologies and ensure that they're being used safely and effectively.
So it's about fostering innovation while also protecting public health.
Absolutely. Another key challenge is the need for more collaboration and data sharing between researchers, industry, and regulatory agencies.
It's all about working together for the greater good.
Exactly. When everyone collaborates and shares information, we can accelerate the development and adoption of those new technologies and ensure that patients have access to the safest and most effective medications possible.
Well said. And with that, we've reached the end of our deep dive into the world of drug impurities.
It's been a fascinating journey.
It really has. We've explored those different types of impurities, the science behind detecting them, the complex world of regulations, and those groundbreaking innovations that are shaping the future of this field.
I hope this deep dive has given you a newfound appreciation for this often overlooked but critically important aspect of medication safety and efficacy.
I know I've learned a lot, and I hope our listeners have too. Remember, knowledge is power armed with this information. You can be a more informed healthcare consumer, ask the right questions, and make educated decisions about the medications you take.
And as new challenges arise, we can be confident that scientists and regulators will continue to push the boundaries of innovation to protect public health.
That's reassuring to know. Thank you for joining us on this deep dive into the often hidden but always crucial world of drug impurities.
We hope you'll join us next time for another fascinating exploration of a new and intriguing topic. Until then, stay curious.